Welcome to Anatomy and Physiology 2 Lab. In this course, we're going to be starting with Respiratory Anatomy. So our first chapter is Unit 22 in the Laboratory Manual on Respiratory Anatomy, and then the next topic will be Respiratory Physiology in the next unit. So when we're talking about anatomy for the respiratory system, we are primarily concerned the thoracic cavity and the head and neck anatomy, where the organs are located for breathing, bringing the oxygen to our airways and to the rest of the body. Now, the primary function of the respiratory system is, again, to allow oxygen from the air to get ultimately into the airways and to diffuse through as gas exchange through in the bloodstream to the hemoglobin, so it can be uptaken eventually by cells, right? Because oxygen is essential for the cell survival. Now, before we can get that oxygen finally to get to the gas exchange stage, we have to go through the multiple different conducting and other airways to allow that air to actually get to the final portion. Now, so there are multiple organs involved here. Primarily, these are obviously the lungs, which are located in the thoracic cavity in the chest. So we have two lungs, right and left lung, and obviously separated by the heart, by the mediastinum and the heart in the middle. Each lung is separated in different lobes. The right lung is separated into three lobes, the upper, middle, and lower lobe, and the left lung is separated into two lobes. Essentially, these anatomical features do not help us understand exactly how the lung works, keeping in mind that internally any of the lobes contain the same material, allowing for the air to be conducted there with the blood vessels and other components to help the lung do its job. So separating the lobes are the different fissures, like horizontal, oblique, called like that. Again, this is more for in terms of for like surgical purposes relevant, or if you're doing something like an autopsy. For us, essentially, just keep in mind when you see these illustrations of the lungs, you're going to see the lobes, you're going to see the fissures as these separating features. Now, the lungs, just like any other organs, are protected by membranes. In this case, they're called pleura or pleural membranes. And we have parietal and visceral pleura. Essentially, parietal is the more kind of outer membrane that protects the lung, and the visceral one is the deeper inner one. Essentially, the only time we should be concerned about the pleura for us is essentially in case of any kind of pathology or abnormalities, like pleural effusions or others, where there's a pathological fluid accumulating and separating these two layers and causing damage or injury to the lung. But overall, in normal physiology, you do not think about them. You just keep in mind that they are just protective layers, just like for any other organs. Now, best way to talk about the anatomy of the lungs is to say, or actually the whole respiratory system, to say that we have the upper respiratory tract and lower respiratory tract. Upper respiratory tract essentially deals with upper portion of this anatomy, so the head and neck anatomy, right, starting with the nostrils, the nose, where the air goes into the body. And the lower respiratory tract deals primarily with what's going on in the chest once you get into the windpipe or the trachea and deal with the lungs and the rest of the structures there. Again, for convenience of kind of memorizing this, just we separate it like that. And that's how we kind of, when we're studying anatomy of the system, we kind of want to have this sort of way to separate them for ease of discussion. Okay, so let's talk about the upper respiratory tract first. Okay, so we have, as the air goes in to the body, enters through the nostrils, right, the nose enters the nasal cavity there, we have lots of blood vessels, it warms up and moistens the air, the different hairs and the mucus secreting glands allow the particles through the mucociliary action, all kinds of debris and pollution, other things that is harmful potentially in the air, to be cleared out, to be removed, and nice, warmer, moist air to be actually inhaled deeper into the system. And so after it passes through the nasal cavity, that air enters the pharynx, which is separated into three portions. Pharynx is essentially that anatomical part that's behind, kind of in the throat. We have the nasopharynx immediately behind the nasal area. We have the oropharynx immediately behind the oral cavity or the mouth, and the laryngopharynx, which is located, again, the lowest portion at the immediately essentially above the voice box or the larynx itself. So you'll see in the illustrations what they look like. Again, this is a fairly small area, but it is quite important because it actually happens to be shared with the digestive system. So when you're eating and swallowing liquids and food, those particles are also passing through there besides the air that you're breathing in. And so especially keep in mind that different histologic appearances of cells will need to change based on what kind of traumatic impact they're undergoing based on what else is happening there. Right? So keep in mind, you should remember already from histology, is that most of the respiratory system is actually covered by the pseudostratified ciliated columnar epithelium, but this is not the kind of epithelium that is best to deal with trauma of swallowing food and liquids. So in that area where food actually be found momentarily as we're swallowing that food, which is the oropharynx area, we actually have a change of epithelium to stratified squamous epithelium, flat cells, multiple layers. Those squamous cells are much more likely to deal properly with swallowing food and liquids as opposed to more delicate cells of these ciliated columnar. Okay, just something to keep in mind here as you're looking at the histology. Other things to note, so in naropharynx we have the goblet cells, we have the mucus producing cells just like we have in other parts of the respiratory system, nothing unique here. And then laryngopharynx essentially continues the regular respiratory epithelium that we're going to encounter pretty much everywhere in the respiratory system. Anytime the epithelium actually switches considerably, I'll be kind of mentioning this to keep us informed about that so that we don't get confused where the cells are actually, important cells are different. Okay, so now that we get to the larynx, the voice box, right? How do we know we have the voice box? We have that large thyroid cartilage on the outer wall of the voice box. If you kind of remove the thyroid cartilage and go deeper inside, we know we have the vocal cords there, the true and false vocal cords. And very importantly, kind of just above that area, before air can enter through the vocal cords, we have this elastic flap of tissue, flap of cartilage called the epiglottis, which protects the airway essentially when you're swallowing liquids or food. You don't want that food to go into the trachea, into the windpipe, and so epiglottis closes quickly over the entryway into the trachea to make sure that the food actually goes into the esophagus and the liquids, and you don't get aspiration of the food into the trachea, right? Which would be very damaging, cause the cough reflex or gagging reflex, and essentially, right? We don't want any kind of food or liquid particles going into our lungs, into our trachea. We only want air, only want oxygen to go there. Again, epiglottis, a flap of tissue closing over the entryway into the larynx, into the trachea, to prevent aspiration of food or liquids. Then once we get into finally the lower respiratory tract, we enter the trachea. The trachea is a hollow tube consisting of different tissues, but most important for us, it has these C-rings of cartilage that keep the trachea always open, always patent, to make sure it doesn't collapse, so that we have continuous supply of air to the lungs. Okay, you're going to see these C-rings of cartilage again. Why are they C-rings? That's because essentially the posterior portion of the cartilage needs to be kind of more open as to allow the esophagus to travel through that area, and there's like food is passing through there, it needs to be kind of more flexible, able to expand. Keep in mind, right, that the cartilage is more kind of a harder material, less flexible. Just as the trachea descends, right, and it needs to obviously separate for the right and left lungs, that separation, or that bifurcation, is called the carina. And as it separates, now it turns from trachea into bronchi. So we have the primary, secondary, tertiary, other kinds of bronchi, right? There's the main stem bronchi. As they separate, you see again in illustrations what they look like. They continue to have those C-rings of cartilage for quite a while until the bronchi become extremely small and no longer need that protective effects of cartilage. So as the bronchi become smaller and smaller, the bronchial tree going through the lungs, they get to about 1 millimeter in diameter or less. They are now called bronchioles, and then eventually become alveolar ducts, and then end in alveoli. These are the air sacs. These are the pouches consisting of simple squamous epithelium where the actual gas exchange occurs. When you think of alveoli, you should think of like bunches of grapes, and if you cut through them under a microscope, you'll see that essentially they're surrounded by capillaries, by blood vessels, where blood is constantly traveling through, getting ready to pick up the oxygen and drop off carbon dioxide and waste gases. And essentially, again, the job of alveoli is for gas exchanges, finally allow that oxygen to diffuse through the thin membranes of the squamous cells into the red blood cells, into hemoglobin, so the blood cells can actually carry the oxygen throughout the body. And so most important part of the lungs, of the entire essential respiratory system, are these alveoli, the gas exchange. Now, keep in mind essentially that the alveoli again are obviously microscopic. You see them on the slides. Again, it should be quite obvious what they're sort of doing based on their appearance. You might see some blood vessels, you might see some other cells. There are different types of cells actually in the alveoli. This goes a little bit beyond what we need to discuss here for our course, but essentially when you look at the illustrations, try to compare the histology, all these different components through the bronchial trees. You're starting out at the top, going all the way down to the final area of the alveoli, where again the gas exchange takes place. Now, before I get into the histology of the respiratory epithelial linings, keep in mind, and other, the rest of the respiratory system, keep in mind again, a very important feature anatomically of the respiratory system is the diaphragm, right? This large muscle that separates anatomically the thoracic from the abdominal cavity. And it's that movement, that contraction and relaxation of the diaphragm that allows the lungs to properly expand and contract and allow the air to be pulled in, kind of the negative pressure, doing inhalation, and then exhaled when getting rid of the carbon dioxide. Right, so diaphragm, again, is not mentioned here on purpose, but you'll see this in the book. You'll see it. We'll talk about it a lot next time in respiratory physiology. Very, very important organ. Again, located at the bottom of the lungs, kind of, if you think of like on top of the liver, so on top of covering the upper digestive system. That muscle, right, is absolutely essential for breathing and is a type of involuntary muscle, smooth muscle, that allows the respiratory system to function properly. Any kind of damage to the diaphragm or paralysis or malfunction of the diaphragm causes significant disruption of the respiratory system, is obviously might cause lethal effects, as without breathing we cannot survive. Right? So very, very important to keep in mind the diaphragm is there and is a critical organ for the respiratory system. Now very briefly, let's go over some of the histological components. Keep in mind that all histology, essentially for hollow organs, is separated into three main layers: mucosa, submucosa, and adventitia, or innermost layer, middle layer, and external layer. Some authors use different terminology here. Again, I'm going to go with this one. I believe this is the easiest one to sort of kind of remember. And so for mucosa, right, that's the actual epithelial cells that come in contact with the oxygen and the air breathing in. Primarily, most of the respiratory epithelium is pseudostratified ciliated columnar epithelium. Again, keep in mind, pseudostratified or false stratified has an appearance of multiple layers of cells, but in reality it's considered essentially as one layer. Every cell touches down into the basement membrane or basal lamina. They are ciliated cells, as cilia has a very good ability to clear away the mucus that's been produced as it's getting connected or kind of attached to the different pollution and debris particles, and the mucociliary action removes those unwanted materials, clears them away so that only nice clean air can actually get deeper into the lungs. Okay. And again, right, obviously they're columnar, they're tall cells, as we all know, right? So when you see that under a microscope. And then the two primary examples where the epithelium actually is different is that one, in alveoli, we have simple squamous epithelium, obviously for easy diffusion of oxygen. And in the oropharynx, we have that stratified squamous epithelium that I already mentioned. For the deeper layers, for the submucosa, we have mostly loose connective tissue. In some areas, we also have serum mucus glands that produce liquid, kind of more liquid mucus that's nice also to help with the mucociliary clearance and overall kind of cleaning out of the respiratory tract of any kind of unwanted material. And not to forget is that we do have hyaline cartilage making up those C rings of cartilage in the trachea. What kind of cartilage do we have in the epiglottis? Right? This would be the elastic type of cartilage, right? Kind of the same one we have in the external ear. Okay. And in the outer layer, we have the adventitia. This is primarily, again, that external layer. So it kind of varies depending on where we are, just like with other anatomical systems. And mostly we have so dense irregular collagen tissue there, but it might contain other components as well, depending on what the organ is like. And make sure to review all this material in the textbook and the lab manual to see the illustrations for all of these organs, where they are located, what they look like in 3 dimensions, and kind of as best as possible. And definitely make sure to look at the histology components for all of these structures.